Alright.
So for our enzyme project, we did DNA Ligase I. My name is Brittany.
And my name is Brittnee. So DNA Ligase I, it was discovered in 1967 by Robert Lehman in his laboratories. Other scientists also were looking at it, such as Gilbert, Richardson, and Hewerts, but it was really Robert Lehman in 1967 that was kind of given credit for finding DNA Ligase I, its function, and its mechanisms. It's denotated, or it's classified as EC6511. 6 stands for the class that it's in, so it is a ligase, so it's within the ligase class. Its other scientific name is polydeoxyribonucleotide synthase, ATP1. We're just going to stick to DNA ligase for the rest of the presentation. All living cells do contain DNA ligase, but it just differs between eukaryotic and prokaryotic cells. Eukaryotic cells use ATP, while prokaryotics use NAD+ for its catalytic ability. So because DNA ligase I uses ATP, we're going to talk about the one for eukaryotic cells. And in humans specifically, DNA ligase I is coded for an L1G1 gene, and we'll talk about its placement on the chromosome and everything like that in the next slide. Next is its functions. It first catalyzes the formation of the phosphodiester bond between the 3' hydroxyl group and the 5' phosphate group. It seals nicks of DNA that take place during DNA replication and repair. And finally, it joins Okazaki fragments on the lagging strand. Alright, some bioinformatics of DNA ligase I. Again, it's found on chromosome 19, denotated by the L1G1 gene. It's located around right here. It's made up of 919 amino acids, and it has 101,736 daltons. It's made up of 3 domains, and those are NTase, OB-fold, and DBD. A little background information on these different domains. NTase and OB-fold are the minimal catalytic unit of the enzyme. They're conserved in all known DNA and RNA ligases, but these 2 domains are the site at which, and they have the active sites and the residues in which DNA will bind. Specifically, they have phenylalanine residues in which the minor groove of the DNA will bind to, and then that binding will be stabilized by aspartate and arginine residues. The DBD domain, it gives most of DNA its binding ability. It makes contact with DNA upstream and downstream of the nick made during DNA replication and repair. And it also stabilizes DNA in its unwound form, so it doesn't become more unwound. All of these domains are connected to each other, so it pretty much makes a ring around the DNA structure during replication and repair. There are cofactors and co-substrates for this enzyme. The cofactors is magnesium 2+, and the co-substrate is ATP. And in a later slide, we're going to talk about kind of their job, where they bind onto the enzyme and what they really do in this process. Finally, their predicted PI for this enzyme is about 5.5.
So after the enzyme is produced, certain post-translational modifications have to occur. And one of the most common ones is the phosphorylation of serine. But each of these marks on this line represents one modified residue. So for serine, for instance, serine 66 is one of the most important ones because it is involved with the replication factories in pCNA, or proliferating cell nuclear agent, which we will talk about in a lot more detail later. And the cyclin binding motif of the OB-fold domain is also involved with the phosphorylation of serine residues. Another one that is phosphorylated is threonine. It's not phosphorylated nearly as much as serine. And then there's also one acetylation of a lysine residue closer to the C terminal of the enzyme. So here's a mechanism of DNA ligase. The reaction always begins with the hydrolysis of ATP to AMP, which is then covalently bound to a lysine residue in the active site near the N terminal. And after that, the adenylated enzyme then transfers the AMP to the 5' phosphate of the DNA backbone where the nick is, and then the 3' OH attacks the 5' phosphate, and that seals the nick. And although there isn't really any conformational change with these enzymes here, there is approximately a 20-degree pivot in the DNA binding domain.
So we talked earlier about how there were cofactors and co-substrates for this enzyme. So first we're going to look at the cofactor of magnesium, 2+. These purple blotches here kind of designate magnesium and kind of where it binds. Magnesium is a metal ion and it has two sites to where it binds. It binds in the active site on the enzyme itself. Its main jobs, the first binding of the first magnesium ion that binds, its job is to stabilize the development of the negative charge on the phosphate of the AMP leaving group. And then the second one is to activate the nucleophilic 3' hydroxyl group's attack on the 5' phosphate group and to create that phosphodiester bond. And then we also have ATP as a co-substrate. ATP's main job is to kind of give the enzyme that catalytic kick to have that, to create that ligation for the phosphodiester bond. Specifically, ATP binds to a lysine in the N-TASE domain, and it is responsible for holding the AMP cofactor deep within the pocket. DNA ligase I does not contain the phosphate residues to give it that catalytic kick, so that's where ATP comes into play. So we talked about kind of the mechanism and the function of DNA ligase I. Now we're going to talk about what happens when DNA ligase I malfunctions. The first thing that we saw was DNA ligase I deficiency. With these individuals, there's an increase in immunodeficiency and an increase in sensitivity to damaging agents such as UV radiation and gamma rays, things such of that nature. There is a delayed growth and there is a poor proliferation of lymphocytes, which kind of ties back to the immunodeficiency. This specific deficiency was only seen in one human, but one human case study, but scientists did go and replicate this deficiency within mice, and they did see these same symptoms. Another syndrome malfunction is Bloom syndrome. This is a autosomal recessive disorder. And these individuals have a predisposition to cancer and genomic instabilities, probably because they have an increase in chromosomal breakage. And so this syndrome is very rare, but it is very debilitating. With individuals with DNA ligase malfunctions, there's an increase incidence of cancer and neurodegeneration. And something else that we found that was very interesting, on residue 566, which is a glutamate, usually if there is a switch out, if lysine comes into play instead, we see very severe immunodeficiency and a delayed processing of Okazaki fragments.
So for our experiment, we found a study that was done in the early 2000s that dealt with the functional complex between DNA ligase and pCNA. There were a lot of facets to this study, and they're all listed here. These are the main ones we focused on, but they all involved proliferating cell nuclear agent, which is pCNA, and this is what the molecule looks like. It has 3 subunits, and they're all the same, so it's a homotrimer, and what it does is it binds to the DNA, and that stimulates DNA ligase to then come and bind as well.
So they did a bunch of different experiments, but the main things that they did within this experiment, we're going to go through the methods. They first used 11 oligonucleotide substrates, and they did 2 main assays, an enzyme assay and electrophoretic mobility shift assay. Within the enzyme assay, they performed the reaction buffer. It was incubated around 37 degrees Celsius, dyed with formaldehyde, heated to around 95 degrees Celsius for about 5 minutes, separated on a polyacrylamide and urated denaturing gel, and then the products were analyzed through a phosphor imager analysis. Then in the electrophoretic mobility shift assay, the reaction was performed in a binding buffer, incubated around 4 degrees Celsius for about 15 minutes, separated on an agarose and a polyacrylamide gel, and then that was also, the products of that assay were analyzed through a phosphor imager analysis. So the first experiment that they did and we found very interesting was how they wanted to look at pCNA stimulating DNA ligase I activity. So within this experiment, and the first gel that we're going to look at over here, gel A, they have in lane 1 just pCNA by itself. Lane 2, they have DNA ligase I by itself, and then lanes 3 through 7, they have DNA ligase I and pCNA, with pCNA's concentration increasing. As you can see, and as the researchers saw, there was an increase in product formation as pCNA's concentration increased, and it was about 5-fold enhancement. So what they saw with that was that pCNA, an increase in its concentration, is increasing the activity of DNA ligase I. And so they found that very interesting, and saw that DNA ligase I must be dependent on pCNA's concentration. In gel B, what it's shown here is pCNA, it works with a lot of different enzymes, and so what these researchers wanted to ask was, is it specified for just, it works with a bunch of enzymes, but is L1G1, is it specified for that one to really enhance its activity, or can it do that with other ligases? So they used the T4 ligase here, and they did the same thing with all rows, increasing pCNA concentration. And what you can really tell here is there wasn't as much product increase as there was here with DNA ligase I. So what that just kind of shows is that pCNA is really specified for enhancing the activity of DNA ligase I. The next experiment we found was interesting within this paper was pCNA enhancing the binding of DNA ligase I to substrates. So what they did within this experiment was, in lane 1, you just have a DNA substrate by itself, 2 pCNA by itself, 3 DNA ligase I by itself, and then 4 through 6 is an increase in pCNA concentration with the DNA substrate and the DNA ligase I enzyme. And so what we see here is there is an increase in DNA complex, which is what they were expecting. So pCNA increases DNA ligase enzyme binding to its DNA substrate. What's happening in lane 7 is they have a high concentration of just DNA ligase I enzyme by itself. And you see that it really creates a high concentration of DNA complex. So what the researchers are kind of getting at in this particular part of the study was DNA ligase I can create a lot of complex by itself at high concentrations, but within our body naturally we do not have high enough concentrations to create this complex. So we need pCNA within our, which does happen at high concentrations in our body, to create these complexes.
So the researchers also looked at the role of phosphorylation, because DNA ligase I is a substrate for an enzyme called casein kinase II. And there are several of these phosphorylation sites near the N-terminal region of DNA ligase. 2 of these sites are serine 66 and serine 141, and they have properties optimal for casein kinase II. Now, the phosphorylation of DNA ligase does not necessarily inactivate the enzyme, but it does prevent the interaction with replication apparatus. So naturally, the researchers wanted to know if phosphorylation of DNA ligase affected the interaction with pCNA. So this gel does not contain casein kinase II, so there's no phosphorylation, and this gel does contain casein kinase II, so there is phosphorylation. And you can see here there isn't really much stimulation or product form, but here you definitely have an enhanced formation of the product. So this demonstrates that phosphorylation of DNA ligase prevents pCNA from stimulating ligation activity. So we also have a couple practical applications using DNA ligase. The first one is peptide antigen sequences, and here's an example of 15 amino acids. 4 of them are negatively charged and 2 of them are positively charged, which makes the isoelectric point about 4. And what peptide antigen sequences do is that they help you generate antibodies. So these are super beneficial because you only need to know the protein sequence, the epitope is well defined from the beginning, and they're really cost effective and pretty easy to synthesize. And the second, we found a study that showed the presence of DNA ligase is higher in tumor cells than in normal cells, and they also found DNA ligase antisense oligonucleotides suppress cell proliferation. So this suggests that DNA ligase plays more of a role in proliferating cells than in resting cells. So what this does is it warrants the design and testing of human DNA ligase inhibitors as new anti-cancer agents. And lastly, genetic engineering. It's pretty well known DNA ligase is heavily used in this field of study because, for instance, with restriction enzymes that cut the DNA backbone, you can just use DNA ligase to glue those ends right back together.
And here are our references. Thank you for watching.